What happens when we tell students to study their math lessons? Most of them say how hard math is for them. Others may give it a shot, but some will have difficulties with numbers. But did you know that there are ways to build and boost your math skills while having fun at the same time? And one of those ways is playing games. Sharpen and speed up your math skills with V-Smart Games Number Sorcerer. Number Sorcerer is a mobile game application developed by Vibal under vSmart to help learners develop and enhance their numeracy skills, particularly in basic mathematical operations. How do you play? It's easy as 1, 2, 3. Simply find the correct answer from the choices within 8 seconds for the easy level, 10 seconds for medium, and 15 seconds for the hard level. Players need to find either the sum, the difference, the product, or the quotient of each numerical expression. Learners will be able to practice addition, subtraction, multiplication, and division. Make sure that you get the right answer so you won't be beaten by the bad guys and not run out of lives. Power-ups are also rewarded when you don't miss a single correct answer in the entire match. When you run out of lives, the game is over. Number Sorcerer can be enjoyed by anyone who wants to practice and improve their math skills. Incorporating digital game-based learning is another intervention for keeping students engaged. That's why mobile games are a great learning supplement because children learn while having fun. Number Sorcerer is now available for free download on Google Play and Apple App Store. This app will soon be integrated into vSmart Learning Management System.
Good day, Kavibal, and welcome to our YouTube webinar series. For the discussion today, the topic will be on DNA barcoding and molecular phylogeny of selected flora and fauna in the Philippines. Before we begin, take note of the following reminders. Make sure you are registered to the webinar to have your e-certificate of participation. Visit certificate.vibalgroup.com to generate your proof of attendance. Share the video using hashtag LearnAs1PH as our official hashtag to all our Vibal webinars. Experience learning ka-Vibal! And now, to proceed with our webinar this afternoon, it is my pleasure to introduce to you our distinguished speaker today. Ms. Lorraine Joyce M. Del Rosario is a faculty member at the University of Santo Tomas Senior High School Science Department and a former bar road instructor at the University of Santo Tomas College of Science. She has been teaching fields of science and practical research and has also been appointed as the moderator of La Stampa, the official publication of the UST SHS, and as an e-learning specialist and educational technology coordinator in the past. She was a member of the UST SHS Executive Committee and some academic-related societies and organizations in the biological field. Apart from graduating as valedictorian in the special program in journalism in high school, Ms. Del Rosario earned a BS Biology degree from the University of the Philippines Baguio in 2017, supported by a DOST scholarship. Her undergraduate thesis with her partner and advisor was awarded as one of the top 2 best paper presentations in the 3rd Philippine Solid and Hazardous Waste Management Conference. She is pursuing her Master of Science degree in Biology at UP Diliman and has been granted with UST HRDP Scholarship in 2021. Her field of interest in research is in genetics, particularly in the study of DNA and molecular phylogenetics and meta-barcoding. Currently, she is part of the UP Diliman group in the Portec project under Philippine Ballast Water and Biofueling Management Research. Aside from being an educator, graduate student, and researcher, Ms. Del Rosario spends her time writing, tutoring English online, speaking and hosting in public, and traveling as a hobby prior to the pandemic. Ladies and gentlemen, let us all welcome Ms. Lorraine Joyce M. Del Rosario.
Good day, everyone, and welcome aboard. The world is a plethora of species, not just containing floras and faunas, but innumerable other organisms who seek their placement in the so-called biodiversity of life. And for several centuries, we aim to preserve and conserve this. Hence, we exert efforts to make each one known in order to determine what they specifically need for survival, adaptation, and evolution. So join me. Welcome to my presentation, my research or science investigatory project presentation, generally entitled DNA Barcoding and Molecular Phylogeny of Selected Flora and Fauna in the Philippines. My name is Lorraine Joyce M. Del Rosario of the UP Diliman and University of Santo Tomas Senior High School. And as always, we would like to thank the Vibal Group for such kind of opportunities in presenting researches that have huge impact to the society. Without further ado, let's begin as I introduce to you the outline of this presentation. So basically, we're going to discuss DNA barcoding of a selected flora, so one representative from the plants, and fauna, another representative from the animals in the Philippines. So specifically, we'll be tackling Philippine hornbills and Argosema species collected in the Philippines. And our presentation still divided into the basic parts, introduction and objectives, methodology, results analysis and discussions, and conclusion. Let's begin with our introduction. Biodiversity describes the variety of species, organisms, and life forms in a given ecosystem. Bacteria, fungi, animals, humans like us, and microorganisms make up these ecosystems. And the variety of species and life forms help maintain a healthy balance within this ecosystem. And for humans, well, biodiversity helps provide food, shelter, medicine, and clean water. It describes all levels of life, different kinds from individual genes to communities of species, to entire ecosystems such as forests, lakes, marines. Biodiversity has sustained life on Earth for billions of years. And actually, it's not evenly distributed across the globe. There are tropical forests covering just less than 10% of the globe and supporting, some world species. Marine biodiversity is highest in the mid-latitudinal regions of the ocean and along the coasts of Western Pacific. But honestly speaking, while many are already known, there are still more which are unknown and which are waiting to be discovered. Hence, scientists have been really exerting efforts to learn more about the world around us, to discover more species. There are a lot of cryptic species everywhere. And we aim to unravel them because they too have a special place in this so-called biodiversity. They have a niche, a role to play in the ecosystem. So what are different ways to measure biodiversity? Well, we have species richness, genetic diversity, ecosystem diversity, endemic species, and unique ecosystems. But this could all boil down to the molecular level. One of them, we go to the starting point, we go to the step one, we can be able to identify organisms with the help of what is known as the deoxyribonucleic acid, the DNA, the blueprint of life. So imagine having a blueprint of life, which is unique per organism, whether you're an animal, you're a plant, you're human. We have different sequences of genes. We have different DNA sequences. Just like when you go to the grocery, you'll find a lot of products there. And how would you know about this certain product? For example, a box of milk. How would you know information about this box of milk? Well, other information which cannot be found in its external factor or in its external appearance. Hence, they have what we call barcode, right? And using a barcode reader and the technology, such as a PC, the data about the milk can be seen. Or if not barcode, nowadays QR codes are common, right? So when we have a simple QR code, when we take a picture of this QR code, we are directed to learning more about what it is. We are directed and it gives us an access to information. So analogically speaking, that is how DNA barcodes also work. When we say DNA barcode, it's a unique pattern of DNA sequence that can identify a species, like the unique patterns in a universal product codes that identify products. So barcoding is a standardized approach to identifying plants, animals, microorganisms, and the like, by even just also minimal sequences of DNA, and we call this DNA barcodes. So these specific short sequences of DNA is unique per organism. And when we have this, when we're able to identify this, then we can identify this organism. So for example, you have a cryptic species. Morphologically speaking, it's difficult to know what it is. And considering in different places around the world, right? These species could be known about something. It has a different common name. It has a different local name. How would we know about it? There has to be a standard method to identify. So using DNA barcode, these short sequences, they form a uniform locality on the genome used for identifying the species. So very interesting, right? Very amazing. So this is one of the greatest advances of DNA technology, which is already applied in innumerable fields, such as species identification, of course, conservation, preservation, and a whole lot more in order to be able to know more about the species. Just like in human, right? We can be able to identify through DNA, we can be able to identify paternal lineages. We can be able to identify suspects of a crime. So because of this unique sequence to each organism, we are brought to greater heights in the field of genetics. So it goes like this, right? So from organism, we can be able to isolate or extract DNA. And of course, with the help of algorithms, with the help of bioinformatics, information and communication technology, or information technology in general, we can generate barcodes. And of course, we can establish databases of these barcodes. So it already currently possesses great potential in identification, confirmation, and classification. Yes, classification of organisms based on DNA sequences that serve as these unique barcodes to each individual species. And actually, in the last decade, DNA barcoding has become an increasingly important tool to catalog the diversity of life and to improve taxonomic classification. We cannot just rely on morphological descriptions, on the external features that we see from organisms, because there's a high likelihood that we could have a mistake in identifying them. So we seek into the molecular level, and that's how we know the true nature, the true identification of the organism or the species. And, species identification, it's an essential issue in the sciences, in casework on biological specimen, because thus having a wide spectrum of potential applications in biodiversity research, conservation, biosecurity, and applied sciences. So let's learn more about DNA barcoding. So you are all well aware that DNA sequences are significant sources of information, right, for improving the understanding of the evolutionary and genetic relationships of organisms. So DNA barcoding, as you can see in our screen, it's a standardized approach to identifying organisms, plants, and animals, as I mentioned, by these minimal sequences of barcode. So these barcodes are gene sequences taken from a standardized portion of the genome that's used to identify the species. So we'll learn more about that in the next few slides. So the use of DNA sequences for species identification has a long history, but sadly, it received significant attention only after it was formally proposed as DNA barcoding in 2003. But to say at present, more and more studies are being unraveled with the help of DNA technology. And without a doubt, it has high impact considerably to the society. I hope we will also gain deeper appreciation of DNA barcoding as we go along in this research presentation. So yeah, what are the benefits? What are the applications of these DNA barcodes or this DNA barcoding in general? Well, of course, it's a taxonomic identification tool. It could be an alternative or additional to morphology. So the best way to identify organism is, of course, DNA barcoding in support to the morphological description. Morphological means the external, right? How we are able to describe the organism based on the features that we can see, we can observe from them. Secondly, of course, it's a very rapid, very quick once you were able to generate barcodes or sequences, you can already subject them to studying more about them using software, and we can be able to create barcodes which we can add or contribute in the databases. And honestly speaking, it's relatively low cost. It's a low cost technique, especially if you're doing bulk, right? Bulk samples. So this can process a great number of specimens at a time. Thus, it is useful, for example, in biodiversity survey. So in the laboratory, we're sequencing thousands of organisms all at the same time. And it's possible with the help of the advancement of technology that we have right now. So in reality, we can be able to identify many, many organisms if we only have the dedication and the passion to do this kind of research work. And of course, once a reference database is already established, it can be applied by non-specialists, right? So it can help even those who are not taxonomists, even those who are not geneticists. It can contribute to our lives. So why do we barcode animals and plant species? Because as you can see in this illustration, known biodiversity, approximately 1.7 million named species of plants and animals. We have the scientific name, the Latin names, in order for them to have a standard name or naming method. However, look at the estimated biodiversity, 10 million. What is the proportion of 1.7 million to 10 million? So there are more to know, more to unravel. And by harnessing advances in electronics and genetics, barcoding will help many people quickly and cheaply recognize known species and retrieve information about them. So the speed discovery of the millions of species yet to be named, it would provide vital new tools for appreciating and managing the Earth's immense and ever-changing biodiversity. So yeah, of course, we're already talking about benefits, right? And we need this for standardization. So we collect samples, we analyze it, we can also compare it. So in order to identify this certain species, we can have a reference of the databases, such as the NCBI. So the NCBI has a collection of many different kinds of sequences of different kinds of organisms. It's being built, it's being established as we go along. And who knows, if you become interested in this kind of research and you were able to discover new species, new sequences, which are not yet available in the GenBank, then who knows, you might just actually contribute the very first sequence of that species. It's already a big achievement, honestly speaking. So like convergence on one or a few railroad gauges, barcoding aims to capture the benefits of standardization, which typically lowers cost and lifts reliability and thus speeds diffusion and use. So for barcoding, standardization should help accelerate construction of a comprehensive, consistent reference library of DNA sequences, speed development of economical technologies for species identification. So the goal is that anyone, anywhere, anytime would be able to identify quickly and accurately the species of a specimen, whatever its condition. And where are these sequences found? Where are these sequences localized? So in the case of most mammals, we use the mitochondrial DNA, and there's a region there known as the COI. So mitochondria, it's an energy-producing organelle in plants and animals, right? And they have their own genome. So we have mitochondrial DNA, we also have nuclear DNA. So mitochondria have their own genome. 20 years of research have established the utility of empty DNA sequences in differentiating among closely related animal species. So 4 properties make mitochondrial genomes specifically suitable for identifying species. Firstly, results so far suggest that a mitochondrial gene will enable identification of most animal species, has been proven in experiments. For plants, mitochondrial genes do not differ sufficiently to distinguish among closely related species. Promising approaches to standardize plant identification use, one or possibly more barcode regions are under development. And of course, mitochondrial DNA is very accessible. So these COI genes, specific segment in the mitochondrial DNA, is very accessible. It's available. All right. And there are a lot of references using this. Therefore, we can easily compare them. So what's COI? Cytochrome C oxidase 1. It contains differences in the representative of those in other protein coding genes. That's why it could be unique per organism. So possibly gains in accuracy or cost using a different protein coding gene would like to be small. So defining a standard region and making sequence comparisons among protein coding genes are easier because they generally lack insertions or deletions present in ribosomal genes. And mitochondrial protein coding genes generally contain more differences. More differences, more variation, easier identification as compared to the ribosomal genes, and thus are more likely to distinguish among closely related species. So why select the barcode sequence from within only one gene? Well, if you're using a lot of genes, working with sequences that straddle genes can actually pose many kinds of problems, and it would be very complicated. It will be a struggle. So within one gene is okay to prevent further complications. And here are also, in other organisms, if not mammals, here are markers which we can use for DNA barcoding, such as RBCL, MATK, 16S rRNA, DNA-K for bacteria, ITS for plants, for fungi, replicase synapsin 1, and a whole lot more. So yeah, that's how DNA barcoding actually works. But later on, when we discuss methodology, we'll be more introduced to how it is in the experiment, how it is from data gathering procedure to laboratory procedure to analysis using bioinformatics, using softwares. And of course, referencing from databases. So in this research presentation, I've selected research studies of DNA barcoding a specific animal and a specific plant in the Philippines. Everyone, the Philippines is a home to a plethora of life, right? And it's actually identified as one of the world's biodiversity hotspots, as it serves a home to a cauldron of flora and fauna. At the same time, it is also listed in the 8 hottest hotspots in terms of numbers of endemic species per area ratios, and habitat loss. On the other hand, however, there are some controversies in DNA barcoding about in the Philippines because of some obstacles, some struggles. And honestly speaking, we also need a lot more experts. We also need a lot more scientists who will be dedicated in doing this. We aim to make a database, a barcode database in the Philippines, especially of organisms which are endemic to us. When I say endemic, they can only be found, locally found here in the Philippines. And actually, we have a lot. So we can identify species in order to learn more about them. They might need help. They might be endangered. They might be on the verge of extinction. Or these organisms could be possibly invasive ones, right? They could possibly be non-indigenous species, and they are introduced in the Philippine biodiversity, and they're just causing harm to our endemic or native species. Therefore, with DNA barcoding, we'll learn more about them. So let's have an introduction on the DNA barcoding of selected Philippine hornbills. So actually, these hornbills, for such a small country like the Philippines, it's really surprising because it supports an astonishing diversity of hornbills. We even have some endemic ones. So it supports an unusually high number of endemic hornbills with at least 11 species and 6 subspecies being generally recognized. And actually, 7 of these Philippine hornbill species are now considered endangered. All have relatively small ranges, with particular forms occurring only on one particular island or group of islands that were formerly connected by land bridges. So all species of Philippine hornbills were already regarded as threatened by the IUCN and the BirdLife International. So the Philippines was distinguished by having many more seriously threatened endemic hornbills than any other country in the world. So it's a good study, right? DNA barcoding of Philippine hornbill is a good study to be able to identify these hornbills, especially if they are endemic to the Philippines. So actually, yeah, Philippines is blessed with so many birds. Avian, our avian culture, our avian are a lot. We have endemic species. We have different kinds of species. Actually, we're very blessed. But as mentioned, hornbills are among the most threatened bird species in the Philippines. So to introduce about hornbills, among some of the Philippine hornbill species are used in this study for the confirmation of their identities. So voucher specimens of hornbill include Rufus Hornbill, like this one, which is known as the Bucerus hydrocorax. So it's locally known as Calao. It's a large species of hornbill with dark rufous plumage, hence the name Rufus. And white tail, very beautiful. Found only in the Philippines, but currently classified as endangered. So this is one of our voucher specimen. When I say voucher specimen, these are preserved specimen that serves as a verifiable and permanent record of wildlife as it preserves as much of the physical remains of an organism as possible. So this Rufus Hornbill has three subspecies. We have the Luzon Rufus Hornbill, Visayan Rufus Hornbill, and Mindanao Rufus Hornbill. So Luzon is characterized by the red bill, while the Visayan and Mindanao have the red bills that are pale yellow, like this one, pale yellow on the distal half. Yeah, so that's Rufus Hornbill. So we have also the Visayan Hornbill, so as mentioned. A new study of endemic and rare Visayan tarictic hornbill, Penelopides panini, on Negros Island, the Philippines, shows that population of this endangered bird can also double the previous estimate of the IUCN. So honestly speaking, their population is also already getting lower and lower. Actually, in the 2001 assessment of IUCN, they estimated that the total number of this tarictic hornbills in the islands of Negros and Panay are very few, 1,800, and this new study nearly finds 2,600 just in Negros alone. So, the population of tarictic hornbills are decreasing alarmingly. And we also have the Palawan hornbill, Anthroceros marche. This is endemic to Palawan and its satellite islands in the Philippines, according to BirdLife International. Although currently no much amount information are still published about it. So we need to study more about them. So in the hornbills, just like what I mentioned, a 648 BP region at the 5' end of the mitochondrial cytochrome oxidase subunit 1 gene, CO1 gene, is commonly used as the DNA barcoding region for most animal taxa, including vertebrates, so including birds. So this region might serve as a DNA barcode for the identification of the Philippine hornbill, commonly using 2% genetic evidence as threshold to the limit species. And this is also very easily recovered, and it provides good resolution, as evidenced by the fact that deep sequence divergences were the rule between 13,000 closely related pairs of animal species. So CO1, COI sequences of the blood tissues of the voucher specimen. So the Philippine hornbills, those tissue that were used were the blood. So were amplified and sequenced for confirmation of their identities. And as a representative of the plant, we'll be using Argostemma collected in the Philippines. So the Philippines also support an astonishing diversity of Rubiaceae. Among families of higher plants or angiosperms, Rubiaceae is the fourth largest in number, comprising about 660 genera, with approximately 13,000 species of terrestrial trees, shrubs, lianas, herbs, and twiners recognized by simple opposite leaves. So it has the largest number of indigenous species and 4 endemic genera among the Philippine dicotyledous plants. So this is Rubiaceae. Perhaps you can see this at your backyard or in your garden. And so it has with its main distribution widespread in the tropics like Philippines. It dominates the layer of vegetation beneath the main canopy of a forest without penetrating it to a great extent. So a wide range of economic importance has raised particular interest of certain Rubiaceae species as they use as major crops, medicine, and a number of ornamental. However, despite these applications, because of its size, the family is not known completely and it's often undertreated. And the classification also remains unstable. So since Merrill's monumental work, a famous author working on Rubiaceae, no updated account of the Philippine Rubiaceae has been done. Therefore, we want to know more about it. So specifically, there's a genus under Rubiaceae, which is a genus of flowering plant. We call them the Argostemma species. So it's the largest paleotropical genus of Argostemma in Southeast Asia. So Southeast Asia is the center of its biodiversity. So foreign species pose highly medicinal attributes, right? And they have been a great interest for evolutionary studies. So in the Philippines, misidentification of the members have been apparent due to their morphological homogeneity, making it difficult and undertreated, according to Alejandro et al. So due to this, most sequences available in the GenBank are limited to species outside the Philippines, most notably Borneo. So we want to identify the species of Argostemma collected in the Philippines. There are already morphological identification. So in the science investigatory project, their molecular identification can be further divulged. So in the Rubiaceae, the internal transcribed region, spacer, ITS, of nuclear ribosomal DNA has been proven useful in delimiting relationships at the species level. So the nrDNA exists in all organisms, and it has a high copy number deemed to be a powerful phylogenetic tool. So this highly variable region with sequences of great importance is one of the most commonly used DNA markers in plants. And it has been recommended, so ITS, this one, has been recommended as a core plant DNA barcode, being an effective universal region for molecular identification, phylogenetic, and DNA barcoding analysis. So it's distinct in answering phylogenetic relationships of angiosperms, and it's attributed to the characteristic of this marker being highly repeated, containing variable regions, flanked by more conserved DNA sequences with universal primers available for PCR amplification. And all in all, when we're able to construct their sequences already, we can have the phylogenetic analysis using the molecular data. So these DNA sequence for genes and amino acids, they are highly reliable and important bioinformatics tool, common in the field of evolutionary biology. So DNA sequencing is popular because a huge amount of sequence can be generated. And when we are able to determine the similarities, differences in their sequences, we can be able to construct phylogenetic trees like this. So it's like constructing a family tree. We want to know which of the organisms are close members of each other, right? Which are relatives of each other. Therefore, it helps us also learn more about them. So since many molecules which have various evolutionary traits are available, it's important to choose the suitable molecule for the phylogenetic analysis of a given lineage. And the importance of phylogenetic analysis lies in its simple manifestation and easy handling of data. So this simple tree represents is a representation of the evolution that makes us to comprehend and represent organisms. So for the study of hornbills, the two objectives are to provide sequences of the CO1 genes of selected hornbill specimens to the GenBank database that will serve as potential DNA barcodes to identify the organism, and to confirm the identity of the different representative of Philippine hornbills based on the CO1 in order to identify their placement within the family. So of course, this study would contribute to the efforts of updating the genetic databases of endangered and endemic Philippine species and the representation of birds in the taxonomic studies within the country. So this is also a way of identifying evolutionary relationships, right, of the Philippine hornbill to other members of the family and the possible implication of the generated relationship. So finally, this study will prove effectiveness of the CO1 gene in elucidating the relationship of birds belonging to the family and identifying samples belonging to the species. And for the Argostemma, so the specimen used in the study is coded as AR002, morphologically identified as Argostemma species. So the study aimed to identify the specimen collected using molecular analysis, confirm the phylogenetic placement of the genus Argostemma in the Rubiaceae family, and prove the effectivity of the ITS, internal transcribed spacer, as a marker in resolving the status of Argostemma. So of course, it will also contribute to the gene database of Argostemma and the representation of plants in the taxonomic studies within the country. So that's for the introduction. Let's proceed with the methodology. So for the methodology, it's a series of experiments that begins with, for instance, in this case, in the DNA barcoding of selected hornbills, it begins with the acquisition of the samples, right? So as mentioned a while ago, we use the blood of the hornbill. So selected blood tissue samples of hornbill acquired from Palawan were sent to the DNA barcoding laboratory of the UP Diliman for extraction and sequence analysis. So there were a total of 7 tissue samples. These ones. Yeah, so these are the voucher specimen. They were tagged with this number and accession code. So the next process is to extract them and amplify the sequence, the unique sequence. So they were subjected to DNA extraction using a procedure, using a kit by Thermo Fisher Scientific, following the manufacturer's protocol. So, and they are also prepared for the amplification using a PCR cocktail mix. And afterwards, they were subjected to DNA sequencing. So DNA was amplified, right, in 25 UL reactions as its component in a PCR master mix. And after that, they were sequenced. So before sequencing, the PCR products were subjected to what we call age or gel electrophoresis. And in the DNA sequencing, molecular bands signifying DNA extracted is sent to sequencing laboratory for the sequences. So the forward and reverse sequences of the samples were subjected to quality clipping and assembly using softwares. And each of their consensus sequences were generated. So there was data editing, right, in order to polish the sequences and to join the forward and the reverse. So the generated sequences will represent the barcode of the organism. And this could be subjected to blast under NCBI in order to identify the closest match species or sequence in the database. So of course, by doing so, we will know the placement of this organism in the phylogenetic analysis, and we will know what is most likely the identification of this organism. And therefore, these sequences can now contribute to the barcode library, which is important and applied in species discovery, community phylogeny, and even ecological or forensics, and a whole lot more. So this is the NCBI BLAST. So it has the basic local alignment search tool. After cleaning the sequences, after joining them, we have the consensus sequence and we can subject it to BLAST. So nucleotide BLAST, we just copy paste the sequence there and they will, so after a few seconds, actually, the closest match of species will show up. All right, and there are percentages of similarities. So it offers BLAST, which allow confirmation of identification of species by showing the percentages of closest match. Hence, we can also create the phylogenetic analysis. So in the study of Argostemma, it's also the same. So of course, sample acquisition is the first step. We were so to be able to collect samples of the plants, right? So the study was based on field observations and examination of herbarium specimens submitted to the University of Santo Tomas Herbarium for accession. So these collected specimens were also compared to other herbarium specimens for morphological classification. So the next step is DNA extraction. So there is just a specific DNA extraction method for plants and also for animals. So it accounts for the molecular analysis already. So they were, for plant, the leaf tissue, they were subjected to liquid nitrogen and grounded to almost powdered form by using mortar and pestle. So DNA isolation and extraction procedure was performed also using plant kits, DNA kits, following the manufacturer's protocol. And for DNA amplification using PCR, in order to obtain optimal results, so the PCR mixture was carefully analyzed. Note, to look at the molecular band signifying the presence of the DNA extracted, it is subjected to agarose gel electrophoresis or AGE, and of course gel purification to isolate them. So this is for the confirmation of bands. And after the run, this amplified DNA are visualized under UV transillumination. So you can see the bands there. So bands indicating the presence of the amplified gene was cut and subjected for gel extraction, which were then submitted to sequencing laboratories to be able to produce sequences like this. So of course, it doesn't end there, because we have to clean the sequences, we have to edit them. So usually they give us the raw files. So we need to treat those raw files, we need to align them, and then we can subject it to the BLAST. So BLAST and sequence alignment, forward and reverse sequences of the samples were subjected to quality clipping and assembly as well, and their consensus sequences are generated and subjected to BLAST. The basic local alignment search tool, you can search about it under NCBI to identify the closest matched species of sequences in the current database. And as for phylogenetic analysis, this one, molecular phylogeny is constructed by aligning sequences of closely related species using software and technique of bioinformatics, hence forming something like this, which will showcase connections or linkages. So that's about it. So I'm sure you're all excited of the results and discussion of this study. So I hope everything is clear so far. We were discussing about DNA barcoding, and then we discussed about methodology, which began with, of course, manual collection of the specimen, the tissue which we will use, where we can extract the DNA, and then subject it to laboratory procedure to be able to extract the DNA. And of course, afterwards, if you already have the sequences, we can analyze them. We can do bioinformatics. We can use different softwares to be able to generate. And then, of course, compare the barcode to the sequences available online. And, we can create phylogenetic analysis. So let's first discuss the result and discussion. Let's first discuss the result of the hornbills, the selected hornbills in the Philippines. So the table you can see here, this is the DNA of the alleged species of hornbills were sequenced, right, using the CO1 forward and reverse primers, and they were assembled. So after they were aligned, in the BLAST search, these are the top results. So this voucher specimen, in the identified BLAST, so these are the top results. So Tarictic. So initially, we don't know the species of this specimen. We just know them as Tarictic hornbill, for instance, by means of morphological classification. But with the help of BLAST, with the help of our molecular analysis, we were able to know about their scientific names. So here are the top results summarized in this table. So as shown in the BLAST result for the CO1 gene region, the voucher specimens are identified as, so this one is Penelopides panini, this one is Bucerus hydrocorax, the fourth one, Acerus waldani, or Penelopides panini, so they're at equal ranking, and then Bucerus hydrocorax for the succeeding ones. So these are the percentage identity for you to assess also. So of course, we also still account for some percentage error in this, but most likely these are the identification of these sequences. So it's amazing, right? So that was the summary, but when we did it one by one, like for example, this first table here, this is the consensus sequence, the result of the similarity. I mean, when you compare it with the close match, when you input the sequence in the BLAST, and then these are the top results. So we can see Penelopides panini on the top, right? With the highest similarity percentage. So a sequence of the complete genome of the same species also appeared in the BLAST result with the same percentage of identity and query cover, therefore aiding in the confirmation of the initially identified voucher specimen. So for the next voucher specimen, it's another Penelopides panini. So we'll just need to analyze also the other numbers, the other quantitative data found here, so that we can learn more about the species. So this table also right here shows the sequence of the Rufus Hornbill when searched in the GenBank, yielding 99% identity and 90% query cover, which is also good. It's a good data already. It's already reliable, I mean, to be able to identify what is the molecular identification of those specific species. So the query cover, if the query cover is low, it could be because either the sequence generated or the sequence in the GenBank is too short. Okay, because we're just using DNA barcodes, right? So we're just amplifying the specific region in their DNA, in their CO1, or sorry, in their mitochondrial DNA, that's the CO1 gene, because that's the specific region wherein there are a lot of differences. So perhaps we're able to generate just short sequences. Sometimes it's not as reliable. Sometimes we're not able to know what species it is. But basically, that's the concept. And hopefully it's clear. So here are other results, other tables or results of the basic local alignment search tool result for Palawan Hornbill 8 and the Rufus Hornbill 9, 10, and 11. So if you can notice, the Rufus Hornbills are the Buceras hydrocorax. And for the Palawan Hornbill, so their Thai could be Acerus waldani or the Penelopides panini. Okay, so right. Hopefully you're gaining deeper appreciation already. Once we have the sequences, we can compare them to available sequences online to confirm the identification of these organisms and therefore help us determine what are these, right? So if we're able, and sometimes we can also, or most of the time we can also contribute our generated consensus sequences. We can submit it to the GenBank, so to this tool, search tool online, and it will be very helpful and useful for other researchers, right? So some researchers in the future, they will also study about hornbills or about Argostemma sp, Argostemma species. Therefore, we can help them and we can nurture research, right? Because we aim to enrich knowledge, we aim to enrich information gathered from research. So that's the result and discussion of the selected hornbill. Therefore, it's successful. We're successful enough to identify with, very high percentage matches of those morphologically identified hornbill and compared it to the molecular identification. So thus proving their real and actual identification. So if you're going to analyze the result for Argostemma, it's the same way as how the hornbills were subjected for analysis and assessment. So here are the blast results for AR002. That's the code for the collected specimen. So it's showing 89% identity to Argostemma brachyanterum isolate. So DNA of the alleged Argostemma sp. So we're still calling them alleged Argostemma sp prior to molecular identification because we were just able to morphologically identify them. Morphologically, we're relying on the features, right? So they were sequenced. So our marker here is ITS instead of CO1. And after the sequences were aligned, they were input in the blast search. And here's the table showing the result. So it has the highest percentage for Argostemma brachyanterum. Well, the GenBank database yields no exact same sequence. Yet, thus the percent identity of the closest species only states 89% identity. So this may account to limited species of Philippine Argostemma available in the GenBank. As such, the native range of Argostemma brachyanterum is actually in North Borneo. So this voucher specimen cannot be claimed as the species mentioned, though, because it is not 100% identical. So we can only confirm the initial morphological idea and thus resolve only at the genus level. Nevertheless, the blast results still aid in the support of the preliminary analysis stating it is an Argostemma sp using morphological methods. And as such, So molecular phylogeny, we're able to construct trees. So we're able, using the sequences generated, we're able to determine its placement in the tree of the genus or the family, right? So this is neighbor joining tree. So this is the AR002, which we are, yeah, this is the AR002. Its placement is in here. So it's close family with other species of Argostemma. So neighbor joining trees generated using ITS portion of the nuclear DNA show the phylogenetic relationship among the studied taxa of family Rubiaceae. So how do you make this phylogenetic tree? We also collect other sequences. Well, we can get them from GenBank. It's free, available sequences. And then there's a software to construct the phylogenetic analysis. So we can align all the sequences there, and the software is already automatic in making the tree. So there are numbers here for likelihood, for percentage values. So the values on the nodes, they show the bootstrap. These are the bootstrap values. Bootstrapping is a resampling analysis that involves taking columns of characters out of your analysis, rebuilding the tree and testing if the same nodes are covered. And this could be done in iterations. So aside from neighbor joining, we can also have the this one, the maximum parsimony tree. It's another type of phylogenetic tree based on criterion or character. And so this is based on criterion and character. As seen from the tree, members of the same genus clustered together, confirming their similarities in characteristics. Hence, they belong to similar taxa. So values on the node still represent bootstrap value. Okay, so AR002, our voucher specimen for Argostemma, still able to cluster with other members of the Argostemma genus with a higher bootstrap value of 89% as compared to what is shown in the NJ3 a while ago, the neighbor joining tree, thereby confirming that it shares the same characteristic. Hence, it really belongs to the clade. Okay, so this is the clade. Normally, being a region of the nuclear ribosomal DNA, the ITS is presumed to have high informative sites, although this may vary, of course, in the types of plants. So in this maximum parsimony tree, there are known characteristic states for a set of taxa or OTUs, and the maximum parsimony method is actually good for similar sequences or a sequence group with small amount of variation, since there is a need to find an order of branching and ancestral configuration of character states requiring the maximum number of character state changes to account for the descent of OTUs. Okay. So additionally, in terms of the markers used, due to its some markers, perhaps some markers when they're applied, they have low resolving capacity. Data generated on the most parsimonious tree would not be considered credible or reliable if that's the case. So in this case, ITS would serve well in combination with other markers. So we can also use other markers to prove other plant markers as for specific taxon study. And when searching for the most parsimonious network that describes a collection of characters, it is necessary to add additional cost considerations. So those are the result and discussion for our representative of a flora and fauna using Dna barcoding. So let's proceed to conclusion and recommendation. So to summarize the result of the study on hornbills, the failure to confirm the identity of one of the voucher specimen there, which is supposedly Palawan hornbill. So if there's failure in identification, it could account for a non-existing CO1 gene in the current GenBank database. So it's not actually all bad news, right? So who knows, maybe you're the first one to have such sequence. So the aligned sequence searched through BLAST match all the morphological modification except in one of the species a while ago, so which is the Palawan hornbill. And now to estimate the reliability of species identification, BLAST method is used. However, it should be taken into account that BLAST analysis only relies on the availability and accuracy of published accession numbers. So of course, if it's not available in the database, we'll also find a hard time learning more about this species. But who knows, it could be a new species, or it could be the first of its own. So the CO1 gene can be a useful marker that can differentiate different species of the Philippine hornbill. And the barcode in general can determine how species relate to each other. So in order for DNA barcoding results to be successful, it is important to be mindful in the step-by-step process that is done from acquisition to laboratory experiments, and of course to bioinformatics analysis. And the effectiveness of the primers also depend in every species, but with their help, it is theoretically possible to observe and exploit variation in the entire genome of species of interest. And of course, PCR rate would depend on different factors such as conditions, cocktail mix, and proper handling. And as a conclusion for the Argostemma study, so ITS, because of the ITS gene, AR002 was acquired from a morphological identified specimen Argostemma. But to estimate its reliability and species identification, BLAST method was applied, and it also proved that it is under Argostemma, although we were not able to determine the specific species. Therefore, in doing so, we could just use the genus. All right, so molecular identification has been shown to be a helpful tool also for species identification, allowing fast, reliable, and cost-effective method. And ITS portion of the nuclear DNA can be a useful marker that can differentiate different species of Rubiaceae. But if we want to be more confident, we can also use other markers. So I guess that's it. For the key takeaway, if developed onto their full potential, history may view DNA barcoding enterprise as one that not only enhance access to taxonomic information, but they can also provide great help with interest in the documentation, understanding, and preservation of biodiversity, which is indeed an exciting prospect. And I just want to share to you this quote. It's also one of the importance of identification of organisms, right? So if you're paying for white tuna and you're eating tilapia, I think you'd want to know that, right? So this is another application of DNA barcoding, which I hope to elaborate to discuss in future VIBAL research presentations, so we can be able to identify organisms so that we are sure of what we are eating or what we are buying, because fraud in the market is very common nowadays. So DNA barcoding will soon enable quick and accurate identification of species of all forms of life. And currently, it's still being developed. So we need more scientists, more experts, more people who will develop interest. So the capacity to blunder slightly is the real marvel of DNA. Without this special attribute, it would still be anaerobic bacteria and there would be no music. So DNA is really, really amazing. With these sequences, we're able to identify species. So that is all. I hope you were able to gain an understanding and appreciation of DNA barcoding, which is my major field of interest in research. Should you have questions, queries, or clarifications, you can always contact me in my email addresses posted on screen. And here are my references, journal references for Philippine Hornbill study and for the Argostemma species study. There we have it. Yeah, thank you very much once again, Vival Group, and hope to see you, hope to discuss to you more research presentations, which would be very interesting and which would really have impact to our society at large. Thank you very much. God bless everyone. Have a good day. Take care and stay safe.
There we have it. In behalf of Vibal Group Incorporated, I would like to thank our speaker for today for this indeed very eye-opening and insightful learning session. It is an honor to have you with us today, ma'am. And to all our Kavibal viewers, all thanks to you for your continuous patronage to all our daily learning session. Don't forget to register to get your e-certificate of participation. The link is in the caption of the webinar. We also encourage you to subscribe and watch on our official Viva Facebook and YouTube channel. Muli, maraming salamat at magandang araw sa ating lahat.